Once the filtrate is formed, the early tubular segments of the nephron reabsorb solutes and water back into the blood to restore its volume and composition. They also remove some solutes from the blood and secrete them into the filtrate to fine-tune the blood's composition. Your goals for learning are to examine the passive and active processes of reabsorption and secretion, to understand how filtrate processing differs in early sections of the tubule, to understand the role of the countercurrent multiplier in forming the medullary osmotic gradient. Here's what you need to know: the structure of the nephron, the definition of passive transport, the definition of primary active transport, the definition of secondary active transport, the definition of osmolarity. To review the structure of the nephron, click the link button. If you use a link button, you can return to the page you started from by clicking the return button. To see definitions of terms, click the bold red words. The relationship of glomerular filtration and tubular reabsorption is something like a parent who cleans a child's room by arbitrarily throwing much of the room's contents into the trash box. That's analogous to glomerular filtration. But as soon as the parent is gone, the child quickly reclaims his favorite things from the box. This rescue of valued substances is like the process of tubular reabsorption of solutes back into the blood. All objects remaining in the trash box will be discarded as waste. To be reabsorbed into the blood, substances in the filtrate must cross the barrier formed by the tubular cells. There are two reabsorption pathways, the transcellular and the paracellular. Most solutes that are reabsorbed use the transcellular pathway. They diffuse or are actively transported through the luminal and basolateral membranes of the tubular cells into the interstitial space and then into the paratubular capillaries. Click the arrow to see an animation of the transcellular pathway. The second pathway is the paracellular one through the tight junctions into the lateral intercellular space. Certain tight junctions are not as tight as the name implies and will allow this pathway while others will not. Click the arrow and follow it through the paracellular pathway. Although most substances use the transcellular pathway, water and certain ions use both paths, especially in the proximal convoluted tubule. Both pathways lead into the interstitial space, then through the endothelium of the paratubular capillaries into the blood. Click either arrow to see its pathway again. The previous topic, glomerular filtration, demonstrated that the force driving the formation of the filtrate is blood pressure within the glomerulus. We now turn to the question of what drives reabsorption, which reclaims the valued substances. Let's begin with the reabsorption of water by diffusion. For each of the following questions, select the correct answer by clicking the appropriate box. How can we cause water to diffuse from the lumen through the tight junctions of these generic tubular cells into the interstitial space? No, this will cause water to enter the tubule from the interstitium, causing further dehydration. Yes, water will move from its higher concentration in the tubule through the tight junctions to its lower concentration in the interstitium. Water will also move through the plasma membranes of the cells that are permeable to water. The next question then is, how can we increase the osmolarity of the interstitium? Yes, transporting sodium into the interstitium will raise its osmolarity. Water will then diffuse from the tubule through the tight junctions and permeable plasma membranes to the interstitium, equilibrating the two osmolarities. No, this will reduce the interstitial osmolarity. The last question is, How will the reduction in the intracellular sodium ion concentration resulting from the basolateral transport affect the activity at the luminal membrane? I'm sorry, this is not the correct answer. Yes, the transport of sodium ions through the basolateral membrane will cause more ions to be reabsorbed through the luminal membrane. To summarize, the transport of sodium ions has two important direct results. 1. The interstitial osmolarity increases, causing water to diffuse out of the tubular lumen. And 2. The lowered intracellular concentration causes additional sodium ions to be reabsorbed through the luminal membrane. This provides more sodium ions to be actively transported and enables the cycle to repeat. In addition, as we will see in the following pages, sodium ion transport enables the reabsorption of most substances in the nephron. We will now begin a tour through the renal tubules. We'll see how the specialized cells in each region accomplish a different stage of filtrate processing. Our first stop is the proximal convoluted tubule, where major reabsorption of valued substances occurs. We'll look first at the activities occurring at the basolateral membrane of the tubular cells. Click the box on the basolateral membrane to start the tour. The reabsorption of many substances from the glomerular filtrate in the PCT depends directly or indirectly on the active reabsorption of the sodium ion. The cellular structure responsible for this process is the sodium-potassium ATPase ion pump located in the basolateral membrane. Orient yourself to the parts of the scene and then click the sodium-potassium pump to activate it. Using energy from ATP, the ion pump carries out primary active transport of sodium ions out of the cell and potassium ions into the cell. As it yields its energy, ATP is converted to ADP and an inorganic phosphate ion, or PI. The sodium-potassium pump is found in the basolateral membrane of many regions of the nephron. Now click the ion pump again to see its effect. Watch what happens to the fluid in the interstitial space as a result of the ion pump's activity. Notice that the interstitial fluid is becoming more concentrated, as indicated by the darker color. The net effect of the ion pump's activity is to decrease the sodium ion concentration inside the cell and increase its concentration outside the cell, thereby increasing the interstitial osmolarity. However, this increased osmolarity around the PCT soon draws water from the tubule, decreasing the interstitial osmolarity again, and together the water and solutes diffuse passively into the peritubular capillaries and are carried away. If you didn't notice the interstitium darkening, repeat the activity by clicking the ion pump. Otherwise, click the basolateral membrane to see additional features. Now notice two additional transmembrane proteins in the basolateral membrane, a potassium ion channel and a glucose carrier molecule. Click either protein to see its activity. The potassium ion channel lets most of the potassium ions diffuse from the tubular cells back into the interstitium. It prevents the sodium-potassium ATPase ion pump from causing potassium ion depletion in the blood or excess accumulation in the cells. Now click the glucose carrier molecule to see its activity. The glucose carrier molecule binds only to glucose and transports it across the basolateral membrane by a passive mechanism called facilitated diffusion. Glucose can move either into or out of the cell depending on its concentration. Since in the situation shown here, its concentration is highest in the cell, glucose moves out of the cell into the interstitium. In addition to the molecules you see here, many other substances also cross the basolateral membrane by similar mechanisms. The luminal membrane of the proximal convoluted tubule contains many transport proteins. Here you see sodium channels, along with the sodium-hydrogen countertransport carrier molecule and 2 sodium-glucose cotransport carrier molecules. The activity of all these channels and carrier molecules depends on sodium-potassium ATPase ion pump activity in the basolateral membrane. Click any of the channels or transporters to see their activity. When you have seen each, click the luminal membrane to see the activity of the entire membrane. The sodium ion concentration of the cytosol has been lowered by the activity of the sodium-potassium pumps in the basolateral membrane. As a result, additional sodium ions move from the high extracellular concentration in the filtrate through the luminal membrane to the low intracellular concentration in the cytosol. They are transported by simple diffusion through sodium channels, and they then move to the basolateral membrane and are pumped into the interstitium, as you have seen. Now click a transport molecule to see its activity, or click the luminal membrane. The cotransport or symport carrier molecules carry both sodium and glucose into the cell. The reabsorption of glucose depends on the movement of a sodium ion down its concentration gradient from high extracellular to low intracellular concentrations. This is an example of secondary active transport. Glucose now moves down its concentration gradient to the basolateral membrane, where it is transported into the interstitium by facilitated diffusion, as seen previously. Now click a sodium channel or a transport molecule to see its activity, or click the luminal membrane. The countertransport or antiport carrier molecule carries a sodium ion into the cell and in exchange secretes a hydrogen ion into the filtrate. Here in the PCT, this activity is dependent on the movement of a sodium ion down its concentration gradient from high extracellular to low intracellular concentrations. This is an example of secondary active transport driven by the primary active transport of the basolateral membrane. The hydrogen ion being secreted here is generated in the cell for acid-base balancing purposes. Now, click a sodium channel or a transport molecule to see their activity, or click the luminal membrane. This animation shows only a few of the molecules that cross the luminal membrane of the PCT. Although not shown here, water and many other solutes diffuse or are actively transported through the luminal membrane for reabsorption back into the blood. For most actively reabsorbed solutes, the amount reabsorbed in the PCT is limited only by the number of available transport carriers for that specific substance. This limit is called the transport maximum, or TM. If the volume of a specific solute in the filtrate exceeds the transport maximum, the excess solute continues to pass unreabsorbed through the tubules and is excreted in the urine. Glucose is vital to the body's normal functioning. However, in hyperglycemia, a condition that accompanies the disease diabetes mellitus, excess glucose accumulates in the blood. What do you think will happen if the number of cotransport molecules is not sufficient to handle an abnormally high concentration of glucose in the filtrate? Click the box next to the correct answer. No, the cotransport molecules change conformation at an invariable rate of speed. Yes, the transport maximum has been exceeded. The extra glucose will be excreted in the urine. No, glucose leaving the proximal convoluted tubule and entering the loop of Henle will not be reabsorbed. No, the number of cotransport carriers is relatively fixed and is not altered by glucose concentration in the filtrate. The concept of transport maximum is much like this factory analogy. A fixed number of workers is matched to the maximum number of sacks of sugar they can remove from a moving conveyor belt. Under normal conditions, all sugar is removed because the number of workers and their rate of work is adequate to remove all of the sacks from the belt. However, if the number of sacks moving down the belt is increased, the workers cannot handle the extra load while working at their normal rate. They will remove only their normal amount, and the excess sugar will end up as waste. What you are now seeing is analogous to the condition of hyperglycemia resulting from diabetes mellitus. Excess sugar in the filtrate exceeds the transporting capacity of the available carriers in the cells of the proximal convoluted tubule. Therefore, some of the sugar is not reabsorbed and so is excreted in the urine. The second reabsorption route, the paracellular pathway, uses the small space between cells as a passageway. The tight junctions of the proximal convoluted tubule are not as tight as their name implies. The increased osmolarity of the fluid in the lateral intercellular spaces resulting from sodium-potassium ATPase ion pump activity causes water to diffuse from the tubular lumen through the paracellular pathway. As this water moves, sodium, chloride, and potassium ions may also follow passively in a process called solvent drag. A large percentage of potassium and chloride ions are reabsorbed in this manner. Click the enlarged tight junction to see this activity. Notice that the flow of water into the lateral intercellular space reduces the osmolarity of the interstitial fluid so that it equilibrates with the osmolarity of the filtrate. Click the tight junction to see this again. To summarize the activities of the proximal convoluted tubule, sodium-potassium ATPase ion pumps in the basolateral membrane drive the reabsorption of water and solutes by increasing the sodium ion concentration of the interstitium. Hydrogen ions are secreted into the filtrate to provide an acid-base balance. Abundant channels and transport molecules in the luminal membrane provide passage for solutes to move down their concentration gradients between the filtrate and the cytosol of the cell. Water and solutes move by bulk flow from the interstitium into peritubular capillaries to complete their reabsorption process. As a result, valued substances have been reclaimed. Approximately 65% of the filtrate is reabsorbed into the peritubular capillaries from the PCT. Included in this reabsorbed volume is virtually 100% of the filtered glucose, amino acids, and any proteins that may have escaped from the glomerulus. As the tubule transitions from the proximal convoluted tubule to the thin descending loop of Henle, it straightens and dips into the medulla, and its epithelium changes from cuboidal to flattened squamous cells. As in the PCT, the cells here are permeable to water, allowing diffusion in response to osmotic forces. The increasingly high osmolarity of the medullary interstitium provides a strong osmotic force that pulls water out of the filtrate. Recall that sodium and chloride ions and most solutes cease to be reabsorbed in this region because the flattened cells lack transport proteins. As a result of these conditions, a great deal of water leaves the filtrate, while solutes are retained. This causes the filtrate to become increasingly concentrated as it passes down the tube. Notice the higher osmolarity of the filtrate at the lower end of the tubule, as shown by the darker color. As the loop of Henle ascends toward the cortex, the thin squamous cells transition to simple cuboidal cells of the thick ascending limb and early distal convoluted tubule. Water permeability of these cells is greatly restricted by tight junctions and by a glycoprotein layer that covers the luminal membrane. Click the box on the luminal membrane to see the activity there. Looking at the luminal membrane of these cells, we see that they lack a brush border in contrast to the cells of the proximal convoluted tubule. However, they do have a limited number of short microvilli containing many ion channels and secondary active transport molecules. Now click the cotransport molecule to see how it differs from the one shown for the PCT. Notice that the secondary active transport molecule is not the same carrier as the sodium glucose cotransporter in the PCT. Instead, this carrier molecule cotransports a potassium ion and 2 chloride ions. However, just as with the sodium glucose cotransporter, it is the sodium ion moving down its concentration gradient that drives this activity. The import of potassium does not increase the cell's potassium concentration because these ions diffuse back into the filtrate through the potassium ion channels in the luminal and basolateral membranes. The imported chloride ions follow the sodium ions through the cytosol to the basolateral membrane. Now let's look at the activities of the basolateral membrane of these cells. These cells possess the same sodium-potassium ATPase ion pumps and potassium channels as do the cells of the proximal convoluted tubule. As seen previously, these pumps generate a sodium ion concentration gradient that drives the reabsorption of other substances through the luminal membrane. Notice also the chloride channel, which allows chloride ions entering from the luminal membrane to follow sodium ions out of the cell. Click a sodium-potassium pump and watch the interstitial osmolarity change. The increasing osmolarity of the interstitial fluid is the result of sodium, chloride, and potassium ions being reabsorbed from the filtrate and transported into the interstitial space. The restricted diffusion of water out of the filtrate, as seen on the previous page and in this pop-out view of the tight junction, prevents equilibration of the filtrate and interstitial fluid. The cells of the ascending limb of the loop of Henle are able to maintain an osmotic difference between these two fluids of approximately 200 milliosmoles. The significance of this reabsorption pattern will be explained later in this topic. In summary, as a result of solutes being removed from the filtrate and water being retained, there are two very important effects. 1. The filtrate becomes increasingly dilute as it moves up the tubule. And 2. The osmolarity of the interstitium is 200 milliosmoles greater than the osmolarity of the filtrate at any given level of the tubule. Notice particularly that this has also formed an osmolarity gradient from the bottom of the interstitium to the top. In this medullary osmotic gradient, the osmolarity of the deeper region is greater than that of the region close to the cortex. Now click and drag a milliosmoles box to compare the osmolarities of the filtrate and interstitial fluid. Then click the forward arrow to see how the activities of both the descending and ascending loop of Henle combine to construct and maintain this medullary osmolarity gradient. To demonstrate the current theory of how the loop of Henle forms the medullary osmotic gradient, here's a simplified view of the cortex and medulla. This theory is called the countercurrent theory because of the opposing flow of filtrate within the two limbs of the loop. The complex interplay of the ascending and descending limbs forms and maintains an interstitial osmolarity with a gradient from approximately 1200 milliosmoles near the bottom of the loop to the normal 300 milliosmoles near the cortex. The gradient formed by this activity is essential for the concentration of urine. To demonstrate how the gradient is formed, we will look first at the role of the ascending limb of the loop and then at that of the descending limb, because the ascending limb creates the conditions necessary for the descending limb to function. Click the ascending limb to begin. The ascending limb of the loop actively transports sodium chloride into the interstitium, increasing its concentration while restricting the diffusion of water. The interstitium surrounding the tubule becomes more concentrated, while the fluid inside becomes more dilute. As the fluid moves up the loop, there is progressively less solute to pump out, so the highest concentrations of solutes are near the bottom of the loop. Next, the role of the descending limb in this process is to provide a continuous concentrated supply of sodium chloride to the ascending limb. The filtrate concentrates as it moves down the descending limb because water diffuses into the higher osmolarity interstitium created by the ascending limb. The solutes remaining in the tube reach a concentration approximately 4 times higher than normal body fluid. The effect is much like what happens in the Great Salt Lake when water evaporates and salt is left behind. The fluid that is left is very salty. This salty filtrate provides a ready supply of sodium chloride for the ascending limb to maintain the osmotic gradient. We must now address the problem of how to keep the solutes in the medullary interstitium from being carried away by the blood. The cells of the medulla require nutrients to function properly. However, blood in a normal capillary flowing through the medulla would also reabsorb and remove massive amounts of solutes from the medullary interstitium. This would quickly weaken the osmotic gradient. To avoid this problem, the capillaries of the vasa recta, as shown here, form hairpin loops, enabling them to function as counter-current exchangers. Click the vasa recta to see its function. Here we see a simplified view of one loop of the vasa recta. Blood in the vasa recta loops delivers nutrients, such as glucose and oxygen, to the cells of the region. At the same time, the loops function as counter-current exchangers of sodium chloride and water. The exchange mechanism works as follows. Blood moving down the descending portion of the vasa recta loop passes through areas of increasing osmolarity. Responding to osmotic pressure, water diffuses out of the blood into the interstitium. At the same time, sodium chloride diffuses into the blood as a result of its higher concentration in the interstitium. If this blood now left the medulla, the sodium chloride would be removed and lost to the osmotic gradient. However, the capillary actually reverses direction, so the conditions also reverse. The viscous blood moves up the ascending portion of the vasa recta through areas of decreasing osmolarity. The blood regains the water by osmosis and loses most of the solutes back into the interstitium. The net result is that the blood has brought nutrients to the cells of the medulla without carrying away extensive amounts of solute, which would weaken the osmotic gradient. Here's a summary of what we've covered. 65% of the filtrate is reabsorbed in the proximal convoluted tubule through active and passive transport processes. The filtrate is concentrated in the descending loop of Henle. Water is lost while solutes are retained. The filtrate is diluted in the ascending loop of Henle. Solutes are lost while water is retained. The asymmetrical pattern of water and sodium chloride reabsorption in the ascending and descending loop of Henle creates an osmotic gradient within the medullary region. The vasa recta circulates blood through the medulla to provide nutrients without removing solutes and weakening the osmotic gradient. To test your knowledge, click the quiz button to go to the self-quiz.